Today we are going to solve Pearson Edexcel International GCSE 91 4B 1 2B. This is the 2B paper of Biology 1 hour 15 minutes paper, Monday 9 June 2025. All right. This is the most latest paper. And before starting the solution of the paper, I will suggest you kindly subscribe my channel, and if you have some question or you want some new video over there, kindly give the comment over there. I will try to make the video as soon as possible. And let's start towards this paper. If we just observe there, this is the 670 marks question paper over there. Let's start this. Advice, remember that read each question carefully before you start to answer it. Write the answers neatly and in good English. Try to answer every question. Check your answer if you have time at the end. These are the best suggestions and I will say key points which you should keep in mind for the solution of this paper over there. Let's start, and best of luck for 13 November 2025 paper over there. All right. Here he is saying some questions must be answered with cross, means if there is a fill in the blanks. If you change your mind, then cut the cross like this from the center over there and then mark your answer in new cross over there. Read the passage below and use the information in the passage of your knowledge to answer the questions that follows. Rivers, the arteries of the world. Okay. Rivers are important supplies of water for drinking, farming, industry, and leisure activities. Like our circulatory system, rivers are essential for transport, and you have a homeostatic effect on nature. The biodiversity in the rivers is such that they are habitat for many different species like fish, etc. And in many countries, rivers are under the threat from human impact, particularly in the release of untreated sewage. Okay, means water is being disturbed over there. Flooding can cause untreated sewage to run into rivers from pipelines, and scientists estimated that in England and Wales during 2022, the sewage was released into rivers for the total time of 3 lakh hours, and this sewage came from 1200 different pipelines. Untreated sewage may contain fertilizers, pesticides, pathogenic bacteria, and pharmaceutical drugs over there. Okay, so here he has shown the photograph. Okay, the photograph shows that the sewage being released into a river. The sewage causes the growth of some shows sewage being released into river, and the sewage causes the growth of something called sewage fungus. Sewage fungus looks like fungus, but it is solid collection of several types of anaerobic bacteria. Sewage fungus is common in rivers that are polluted with untreated sewage or near the cattle and intense crop farms. Okay, if the sewage fungus is found in a river, often indicates that the biodiversity of the river will be low. Okay, pesticides from the agriculture also released into rivers, and these are frequently transferred through the natural food chains. Okay, even pharmaceutical drugs such as those containing the hormones estrogen and progesterone have been found in polluted rivers. These drugs must have been consumed by humans, excreted, and then released into the sewage flowing into water. Untreated sewage can also contain pathogenic species of bacteria that then enter rivers. If humans are infected with these bacteria, the large intestine absorbs less water, causing diarrhea. Some of the bacteria are found in sewage are resistant to antibiotics. Okay. Scientists are looking for the ways to prevent river pollution. One way to reduce the risk of pollution from fertilizers and from the cattle urine and feces. So, wood chip bioreactors are being drilled in agricultural fields that are near to the rivers, and these bioreactors are pits filled with the wood chips and denitrifying bacteria. Water drains through these bioreactors, okay, and this helps to remove the nitrates before they reach the rivers, okay. So that is sustainability related to this. In some countries, efforts are being made to replant forests okay, in areas upstream of rivers to reduce the river flooding and the risk of untreated sewage release. Okay, let's move towards the main information which he has given us over there. So he is saying to the questions moving over there after reading the whole passages. River have a homeostatic effect on nature. This is similar to the role of homeostatic in humans. State what is meant by homeostasis. So homeostasis means the maintenance of a constant internal internal environment over there. If I go for explanation, I will say we keep our body temperature constant, we keep our sugar level constant, we keep our hormone release and water absorption up to constant level, so that there is optimum pH, optimum temperature is going to work for the proper functioning of the whole body. So for the environment, same scenario will be done with the help of rivers over there. So he said what is homeostasis. I will say the maintenance of a constant internal environment even in the atmosphere or in the human body over there. Now he is saying calculate the mean number of hours that each pipeline released sewage into rivers over there. So if you remember, total hours were 3 lakh. 1 2 1 2 3, and number of pipelines were 1200. So if I take the average over there or mean over there, then I will say it will be 3 1 2 3 4 5 divided by 1200. Let's go with it. Fraction introduced, 3 1 2 3 4 5 divided by 1200, it will give you 250. So mean number will be 250 hours over there. Let's move to the next part over there where he is saying uh, sewage fungus is not a fungus but is made from several different species of bacteria. Which of these structures are present both in fungi and bacteria over there? So I will say here that it is cell wall and cytoplasm. Yeah, he is right that in bacteria, cell wall is made up of chitin, and in fungi, it is made up of chitin, and in bacteria, it is made up of peptidoglycan over there. But cell wall is present over there, and cytoplasm is also present over there. So I will have to go with option A. Cell wall and nucleus only. Nucleus is not present in the bacteria. Cell wall, cytoplasm, and nucleus. Nucleus is not present in the bacteria. Nucleus is present not in the bacteria over there. Okay, explain why the rivers near to farms may have low biodiversity. Okay, so here I will say that if I go for the reasons for less biodiversity, I will have to give at least 4 reasons for this. I will go with the fertilizers from farms. Fertilizers from farms. Fertilizers from farms can leach into waters. Leach into waters. Remember that marking scheme or true answer sometimes will be different from my answer, because I try to give the concept basically so that you can solve the paper exactly. You can understand the paper. I'm not copying the marking scheme or like that. Okay, let's go with the second reason over there. Like this provides this provides extra nutrients Like I can say it can provide nitrates and phosphates for algae and aquatic plants like that. And aquatic plants over there. So biodiversity is not near the uh, rivers where farms are very near to it. After that, I can say that this leads to a rapid algal growth. This leads to, when the nutrients are too much, this leads to rapid algal growth. And I can say here, then when it is going for rapid algal growth over there, then we can say eutrophication can happen, forming a surface layer that blocks light, forming the surface layer that blocks the sunlight over there. Okay, if I go with the next reason, then I will say here that plants below the surface die. Plants below the surface will die because they will not be able to get light for photosynthesis over there and for living over there. And then I will say that decomposing bacteria will multiply and consuming oxygen during decomposition. If I go more detail over there, then I will say that resulting low oxygen levels cause the death of aerobic organisms like fish and invertebrates, reducing the biodiversity. So this was your four reasons I have written for you, and much more to more reasons which I told you here, that decomposing bacteria will multiply like consuming oxygen during decomposition like that, and the resulting low oxygen levels cause the death of aerobic organisms like fish and invertebrates, reducing the biodiversity near the farms over there. Now he is saying estrogen and progesterone in polluted water lower the levels of FSH and LH in mammals. Okay, state why lower level of FSH can reduce the fertility in mammals. So FSH is basically needed for the development of follicles in the ovaries. If it will be reduced, then fertility level in mammals will be reduced over there. So I will say here FSH is needed for the development of for the development of follicles over there, and follicles in the ovaries, which will reduce the fertility rate. Even the sperms are coming, no follicles are present over there in the ovaries, and no fertilization is going to happen over there. Okay, but he is saying state why the lower levels of LH can reduce the fertility in mammals. So for this one, I will say LH is needed to trigger the ovulation. LH is needed to trigger the ovulation over there. And when it is not triggered, the release of an egg from the ovary will not happen, and this will reduce the fertility in mammals over there. Now he is saying pathogenic bacteria produce poisons that prevent the activity of transport of salt into the blood vessels surrounding the intestine. Explain why preventing the transport of salt into the blood vessel will cause more water to be present in feces over there. So for this one, I will say here that water moves Due to osmosis over there, water moves by osmosis there. By osmosis from intestine into blood, from intestine into blood over there. And if the concentration of salt, if the concentration of salt will be is lower in the blood, is lower in the blood there, then I will say here that the concentration gradient is reversed, the concentration gradient is reduced, the concentration gradient is reduced, and because of this, less water is absorbed. So, less water will be absorbed, and we can say here that from the intestine into blood by osmosis, so more water remains in the feces causing diarrhea. So, more water in feces causes diarrhea. Water in feces causes diarrhea. In feces causes diarrhea over there. All right, let's move to the second part. What he is saying over there, he is saying that explain why overuse of antibiotics has led to an increase in antibiotic resistant bacteria over there. So I will say here that in a population of bacteria, he is asking for 3 marks at least. I will have to go with 3 reasons at least. In a population of bacteria, there is a natural variation in bacteria population. Bacteria population. Uh, there is a natural variation. There is a natural variation over there, and so some individuals may have a mutation, may have a mutation, may have mutation that makes them resistant, that make them resistant and resistant to an antibiotic, to an antibiotic biotic over there. This was the main reason that the genetic variation is happening. And when antibiotics are overused, they act as a selection pressure, so killing non-resistant bacteria. So he is saying when antibiotics antibiotics is overused, antibiotics is overused, then we can say overused, they act as selection pressure, they act as selection pressure, killing the non-resistant bacteria over there. So I will say here that when the non-resistant bacteria are killed completely, then I will say the resistant bacteria, which are the resistant bacteria which survive, bacteria which survive there, Then I will say the bacteria which survive definitely will reproduce and passing to the resistance gene to their offsprings over there. So I will say here the resistant bacteria which survive will reproduce produce to their offsprings, and that will be more resistant over there. So what he was asking over there, explain why overuse of antibiotics has led to an increase in antibiotic resistant bacteria, because only they survive and they have so much food and over there in your body environment, and they can easily go for multiplying over there by using binary fission phenomenon over there of asexual reproduction. Explain how wood chip bioreactors reduce the pollution in the rivers over there. Okay, then I will say bioreactors contain denitrifying bacteria. They contain denitrifying nitrifying bacteria into them. So I will say they are helping to reduce the pollution in the rivers, and these bacteria converts the nitrates. What I say, these bacteria convert the nitrates nitrates into nitrogen gas there. Okay, so these nitrates will not be available to be added into water over there, and nitrogen gas will be released into atmosphere, reducing the nitrate concentration in the water that reaches to the river. After that, he is saying explain why replanting forest in areas upstream of river will reduce flooding over there. Yeah, this is, if you have done the geography paper, you can easily explain this, because tree roots absorb water from soil, absorb water from soil, and and help to create a more uh, porous soil structure, porous soil structure over there, and this increases the infiltration rainwater into the soil, reducing the amount of surface runoff. And with less surface runoff and with less surface runoff, I can say over there that less water enters the river channels simultaneously, so it reducing the effect of flooding over there. Let's move to the next question. What he is saying over there, he is saying that the lungs and the kidneys are excretory organs. Which substances are excreted by the lungs over there? Then I will say that carbon dioxide and urea is not done by lungs. Carbon dioxide and water, this can be true. Carbon dioxide, urea, no. Urea and water, no. So option B is right over there. You will fill it like this. And if you fill something wrong like this, then cut it like this, okay? The graph shows how the volume of air in the lungs changes over time. Volume of the air in the gas and time. So X and Y 2 points are given. Explain the change in volume of air in the lungs from the point X to the point Y over there. So I will say here that the volume of air, the volume of air from X to Y decreases from X to Y, X to Y And this process is called, like take in air is called inhalation, and take out of the gases like carbon dioxide is called exhalation or expiration. So I will say it is exhalation. Spell can be, there will be a silly spell mistake, just ignore that. Exhalation. And the diaphragm and intercostal muscles relax over there. And if you go for more decrease, so I will say that decrease the volume, the this decreases the volume, decreases the volume of the thorax. And I will say here increasing the pressure, increasing the pressure, increasing the pressure over there, inside the lungs, inside the lungs over there. And you can write more about this above the atmospheric pressure, above the atmospheric pressure, forcing air out over there, forcing air out over there. All right, let's move to the next part. He is saying people with a condition called diabetes often have very high blood sugars over there. And he is saying urine from the people with diabetes attracts ants, but urine people without diabetes don't attract ants. Ants are insects that are attracted to glucose. Explain why people with diabetes produce urine that attracts ants over there. So I will say here that the people with diabetes have high blood glucose level, high blood glucose level over there, and this exceeds the renal threshold, the renal threshold over there. Renal threshold is going to increase, meaning which means that meaning that which means that that not all glucose can be absorbed by the kidneys over there, and therefore, so glucose is present in larger quantity, or I can say is present in the urine, in the urine over there. And we can say so ants are attracted towards this glucose in the urine over there. Okay, let's move to question number 3. There was a bad luck that the estimate paper which I have given into the as a guest paper for you, this question was missed by me in the form of guess over there. And remaining all 90% guess was perfect. So I will say sorry for this. Next time I will try more accuracy, inshallah. Okay. Uh, Here some structures P, Q, R, okay, and S are given. Okay. Which structure is the retina over there? Which structure is the retina over there? So that is retina. This diagram shows the labeled structures of which structure is the retina. Retina is related to the outer surface over there. So I will say here that it is S. So S is the retina over there. Then he is asking a teacher investigate the effect of light intensity on the pupils of diameter. On pupil diameter, retina will be, it is P. It is P, sorry. It's P. It should go with A. Let's check which one, but it is P, I think so. But the right one is D for S over there. Okay, let's go to question number B part. A teacher investigate cover the student's eye with a mask. C. Set the light intensity of the room to a low level. Remove the mask. Wait for 20 seconds. Record the image of student's pupil and iris with camera. Measures the diameter of the pupil with the image using a ruler. Repeat two more times and same light intensity is given. Okay. The teacher repeats the experiment at different light intensities. The table shows the teacher's result over there. Okay, 8.1, 8.4, 7.8. Okay, mean he is taking. So you can fill up by taking the mean 8.1, fraction introduced 8.1 plus 8.4 plus 8.2 divided by 3, that is equal to this, which is equal to 8.233. 8.233 dot 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 like millimeters over there. So he said mean diameter of intensity 1 to absorb per unit. One arbitrary unit can give your answer up to two significant figures. So I will go with 8.2 over there. Okay, it is 8.2. I have given it up to two significant figures. Then he is saying, with reference to the data in the table, describe the effect of increasing light intensity on the diameter of the pupil. So I will say here that from 1 to 4. Okay, then it is, I will say as the light intensity as the light intensity increases from 1 to 4, increases from 1 to 4 over there, arbitrary units, 1 to 4 arbitrary units. I will say the mean pupil diameter, the mean pupil diameter increases or decreases, increases or it decreases, decreases over there. And what he was saying, the second one over there with reference table, describe the effect of increasing light intensity on the diameter of the pupil. It decreases. And from the second section, from 4 to 5, the diameter remains approximately constant, approximately constant, like 2.1, 2.2, like that. Constant, if you can observe there, that is 2.1, 2.1, 1.9, 2.0. It is approximately equal. And the in number 4 is also approximately equal over there, or approximately constant. at its minimum size. At its minimum size over there. Explain the effect of increasing light intensity on the diameter of the pupil over there. Then I will say here, in bright light, the circular muscles uh, in the iris contract, and this causes the pupil of eye over there, of eye uh, mm, to constrict, to get smaller, like that, to get smaller or to constrict over there, and this reduces the amount of light, the amount of light entering the eye, which we can say entering the light to the eye, and preventing the damage. So it is preventing the damage to the retina over there, where image will form in the form of, we can say, accommodation over there. State one variable that the teacher should control over there. So I will say the distance between the camera light source and the student eye. He must control, as a variable. Teacher should control the between camera distance between camera or light source from student eye over there. Explain how teacher's method ensures that the measurement of the pupil diameter are accurate. Okay, so I will say here, to get the accuracy, I am saying the teacher is getting the mean values of diameter. So it is the good method to get more precision and accuracy over there. And I will say a mean is then calculated, which reduces the effect of random errors and anomalies and gives more reliable values over there. And mean gives more reliable results or values. Okay. Let's move to question number 4. Here he is saying the photograph shows Prometi, the first cloned horse. Okay. And okay, here he is saying that Prometi was produced by cloning using a nucleus of a skin cell of a horse called Stella comata. Complete a passage of cloning between them. Okay. Here he is saying that dash nucleus was taken from this, that will be definitely diploid, because body cell is always diploid. Nucleus was taken from the stella comata, and this nucleus was inserted into an inculated egg cell or ovum, like that, egg cell or ovum over there, that had been taken from a donor horse. And electric shock an electric shock was used to start the type of cell division, uh, cell division called mitosis, eventually producing an embryo. The embryo was then placed into the uterus of mother. Uterus. Uterus, what the spells I am writing over there? Into uterus. Yeah, or womb. Uterus. 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 Or womb of mother can be written over there. Womb. Okay, that is womb. WOMB. Okay, this was a little bit fill in the blanks over there. Then he is saying until recently, most cloning has been attempted with farm animals. The table shows the information about the success rates of cloning different animals during over there. Okay, and he has saying that cattle 7,500, live bird 75, surviving at least 2 years 55, sheep live birds 50 for 15 sheep over there, goat 525, only 5 survived. Some people now want some people now want to use the cloning to replace their pets. The cost of cloning a pet is much as 50,000 United States dollars. Comment on the use of cloning to replace pets. Use the information in the table and your own knowledge about variation to support your answer. Okay, I'm not writing this answer, just going to trying to explain over there by looking at this graph. Just listen very carefully over there. I will say that the success rate of cloning is very low if you observe there. And the table shows that the sheep, only 50 live births are achieved from 3,500 embryos. So the success rate is almost, it is, I think so, 1.4, 1.5%. There is also a high mortality rate as shown by the number of animals that do not survive 2 years, because like if you observe for sheep over there, only 15 survived out of 50 sheep that were got the live birds over there. After that, cloning is very expensive, as stated, like we observe 50,000 US dollars over there. And a clone, while genetically identical, will not be an exact replica of the original pet in terms of personality or behavior. This is because these traits are also influenced by the environment and experiences. Therefore, using a cloning to replace pets, I will say, is an expensive process with a very low chances of success and will not guarantee a pet with the same character. I think so. This explanation, if you try to write from the video, as I have explained for you, you can rewind the video. Otherwise, I think so. You have got the concept and you know how to go for creative writing about these type of informations. You can easily do over there. Please don't mind. I'm not writing this question answer over there right now, as I have explained you in detail over there. If you want to write for yourself, just listen the video again and note down over there. Okay, W like epidermis, X like the midrib or vein over there, Y like that. Okay, let's go for it. And Z is given also. Which area of the leaf contains the xylem vessel over there? Xylem vessel. Okay, that will be in X over there. So it is option B over there, like it is midrib like that. Okay, X will be given over there. That the xylem is present. If you just look again, this is the xylem vessels to be present over there. Okay. Now he is saying a student uses the apparatus to measure the water uptake by the cut of plant shoots over there. Water uptake is observed by capillary tube and air bubbles over there. The inside capillary tube has slender shape. The air bubbles is length is 5.2 centimeter in over there. And we can say here Okay, the inside the capillary tube has a radius of 0.50 millimeter. Okay, and calculate the rate of water uptake in millimeter cube per minute by the cut of plant shoot over there. Volume of cylinder is pi r square L over there. All right. So we will have to go for the calculation of rate of water uptake. For this one, I will say first of all the radius, which is given 0.50 millimeter over there. And then he said, as we know, the cross-sectional area can be calculated by cross-sectional area is equal to pi r square. So pi 3.14 multiply 0.50 square is being taken over there. Let's go for this. Pi multiply 0.50 square equals to this is equal to 0.785 millimeter square, and like distance moved by bubble will be 5.2. So I will say here that volume for this one will, for uptake, I will have to just multiply this by 52 millimeter to get my volume over there. Just multiply this by 52. So I will have this equal to 40.84. 40.84. Okay. 40.84 there millimeter cube. Okay, now I will have to go for time taken to calculate the time. As he given the time over there, a student cupcake. Okay, what he said over there. The inside of the radius is this, and the time, as he given the time, the inside capillary tube, the bubbles 5 minutes. Okay, time is given here, that is 5 minutes. So I will have to divide the volume by time over there. So divided by 5, because he needs only in per minute. If he needs in per seconds, then I will also multiply 5 with 60. But here he is asking only per minute. So I will divide it by 5. So I will get answer this. That is 8.168 divided by 5 is equal to 8.16 it was. Yeah, 168. So I will say it is 8.2 millimeter cube per minute will be the volume of the rate over there. All right, this was volume rate which he was asking for capillary tube over there. Describe how the student can use the apparatus to investigate the effect of changing wind speed on water uptake by the cut plant shoot over there. All right, so he will have to be sent that set up the apparatus as shown in figure above, and here I will say that use a fan at a fixed and low speed and up and point it towards plant shoot. Okay, and here I will say if it is pointing towards shoot, then he will have to measure the distance, then measure the distance, measure the distance, the air bubble moves moves in a fixed time, which I can say is 5 minutes, like that. Fixed time, like 5 minutes, and calculate the water uptake over there, and repeat the measurement for 2 times at the same wind speed to calculate the mean rate. So calculate mean rate of two values, rate of two values, so that he can get more precise measurement over there. Then change the speed means from the slow it should increase over there. So change the speed, and by adjusting the fan setting or repeating the process each time, ensure that all other values or variables are controlled, like light intensity, or like temperature, or humidity, or type of plant should remain constant over there. So this will be the technique how a student can use the apparatus to investigate the effect of changing wind speed on water uptake by the cut shoot plant over there. Okay, let's move to the next part over there. What is he saying there? You can also draw the figure here, okay? Okay, a scientist investigates how ABA, an auxin-like substance, affects the plant growth. The scientist does not water a plant for 4 days. Then they then water the plant on day 5. Okay. Day 1, day 2, day 3, day 4. ABA concentration in micrograms per gram of a leaf is increasing, and then it is decreasing again. And graph 1 and the graph 2 shows the changes in the percentage of relative water loss, like they are not watering the plant, so at day 4 and 5 he is saying that it is decreasing. On graph 2, comment on the changes in ABA concentration and water loss in the scientist investigation. Use information from both of these graphs. If we look again, what I am saying, that ABA concentration is increasing, and add the day number 2 over there. Okay, and add the number day of 2, percentage water loss is because when at day 2 the water is completely lost again, decreasing the percentage over there. All right, let's go for this. I will say that from day 0, from day 0 to 4, we will say when the plant is not watered, the ABA concentration increases steadily, increases steadily. After that, I can say over the same period, over the what happened with water over the same period, I can say the percentage water loss, water loss, percentage water loss also increases. And here we can say that if water loss is increases on day 5, on day 5, when plant is watered, plant is watered, Third, then I can say here the ABA concentration, ABA concentration drops rapidly over there or sharply over there, rapidly, and correspondingly the water loss also decreases sharply, and water loss also falls rapidly or sharply over there. So in more detail, I can say this shows a strong positive correlation. It shows positive correlation between ABA and water. So when ABA decreases, water loss decreases. When ABA decreases, water loss also decreases over there. Okay. Let's move to question number 6. Apples contains an enzyme called phenol oxidase. When apple's tissue is exposed to oxygen in the air, the enzyme turns apple tissue brown. Okay. A student uses this method to investigate the effect of pH on the time taken for the apple tissue to turn brown. Much apple is produced pulp. Place 25 gram of this pulp into beaker. Mix 7 pH. Okay. Check the color of the pulp every 5 minutes. Record the time when the pulp turns brown. Okay. The student repeats this method using pH buffers 3, 5, 9, and 11. So 3, 5, 7, 9, and 11 he has used over there. Calculate the percentage increase in the time taken for the apple to pulp to turn the brown at pH 11 compared with the time with pH 7 over there. So at 11, it is going to be pulp to be taken as 75 minutes. Okay. So I will say here that it is changing for paper water to time at pH 7. Time at pH 7 will be taken like 5 minutes, and time at pH 11 is 75 minutes. So if I take the difference, that how much the increase over there, then I will say 75 minus 5 is equal to 70. So I will say here that the original was 5. So I will say here that 70 divided by 5 can be taken as multiply by 100, and I will say 5 with 14. So it is the increase over there is approximately 5 and 75. 70 divided by 5, the original one, multiply by 100. So I have taken it as 1400% is the increase over there. All right. After that, he is saying that explain why the change in pH affects the time taken for the apple pulp to turn brown over there. So I will say that the browning is caused by enzyme named we can say phenol oxidase. Phenol oxidase is an enzyme over there, and I can say here that enzymes have an optimum pH at which their active sites are in correct shape for the substrate. If the pH is above or below the optimum, then the active site denatures are changing. So I will say there is optimum pH over there for enzyme. If that is available, then that is good. This means that the substrate can no longer bind, and rate of reaction decreases and takes longer for the browning to occur over there. I think so the reason behind it is much clarified towards you. After that, he saying explain how the students could modify this method to give more accurate measure for of time taken of pulp to turn brown. Over there, I will say that check the pulp color, pulp color frequently, like every minute instead of every 5 minutes. Check the pulp color every minute over there. Then you can get exactly uh, the more modified method for accurate measurement of time taken for the pulp to getting browning over there, and use a calorimeter to the measure the color change. Colorimeter measure the color change objectively. Lee over there, rather than relying on subjective visual judgment over there. Okay, it is. You should use colorimeter as a device for this section over there. A genetically modified variety of apple has been produced that does not turn brown when exposed to air. These apples have a gene in their DNA that produces a section of messenger RNA with the complementary sequence of messenger RNA. Okay, state two differences between DNA and RNA over there. That is too much simple, I think. So DNA is double helix, double helix stranded or double stranded over there, double stranded, while RNA is single stranded. Stranded. After that, I can also say that DNA contains deoxyribose sugar. DNA contains deoxyribose sugar, while RNA contains only the ribose sugar, while RNA contains ribose sugar there. After that, explain why the GM, genetically modified apples, do not turn brown when the apple tissue is exposed to air. Use your knowledge of protein synthesis to support your answer. Okay, then I will say that when it is genetically modified, then I can say the inserted gene produces a section of messenger RNA that is complementary to the phenol oxidase enzyme over there, RNA over there. So I will say that this complementary mRNA, this complementary RNA, uh, this complementary RNA is, I will say, is antisense RNA, where we can say that the antisense RNA, it binds to sense mRNA of phenol oxidase over there, and by base pairing like that, and this prevents the mRNA this prevents the mRNA from being, we can say, translated like that. Translation phenomenon, you remember that, being translated over there, and by the ribosome over there, and I will say therefore the phenol oxidase enzyme is not produced over there, and the Browning reaction cannot occur. All right. Okay. This was your whole paper, which I tried to explain as much possible. And best of luck for your exam. This was the solution of this paper. In detail, the other papers will also be uploaded. And kindly subscribe my channel. If you have some suggestion or you need some video of some paper over there, kindly give the comments, and also share the video and comment the video with your friends, and also ask to your classmates to subscribe my channel so that they can also get the proper benefit from these videos over there. This is just help for the students who are going to do as a private candidate over there. Thank you so much for today. It was the Bio 2B. I will also give the guest paper over there, guest paper video like I did for 1B over there. It will also be going for 2B, inshallah. Thank you so much for today.